皆さん、こんにちは。大変お待たせしました。時間になりましたので、AnaBios Corporation のウェビナーを開始いたします。私は本日のウェビナーのモデレーターを務めます、AnaBios Corporation の日本およびアジアパシフィックでのビジネスデベロップメントを担当しております、林隆と申します。どうぞよろしくお願い申し上げます。まず、AnaBios Corporation についてですが、AnaBios 社はアメリカ合衆国カリフォルニア州サンディエゴに拠点を置くユニークな CRO であり、またバイオテクノロジー企業です。私たちは倫理的に同意を得た健常または患者ドナー様から人臓器を回収し、その組織や細胞を用いて創薬における前臨床化合物を試験するための生理学的圧生を実施しており、本質的にファーストインヒューマンを再定義しています。また、アナバイオス社とのコラボレーションでは、プライマリーな人組織や細胞を用いますので、実際に臨床試験を行うことなく、臨床試験データを予測し得る人 EX-VIVO 試験の実施が可能です。次に、本日のセミナーと演者を紹介いたします。本日は、アナバイオス社のリサーチ&デベロップメント担当バイスプレジデントであります、ナジャ・アビゲルゲス博士により、Human Ex-Vivo Models for Drug Discovery と題して、創薬プログラムをサポートするためのアナバイオス社によるユニークな成人人 Ex-Vivo モデルの応用について紹介いたします。演者のナジャ・アビゲルゲス先生は、フランスのポワティエ大学で生理学、薬理学の修士号を、そして、フランスのパリ第11大学で心臓生理学の博士号を取得しております。アナバイオス社入社以前は、アストラゼネカで創薬プログラムに携わっておりました。製薬業界で17年以上の経験を持つアビゲルゲス先生は、革新的なリーダーであり、抗がん剤、タグリストなど医薬品研究開発とそれらの承認、心臓生理学および薬理学の複数の分野にわたる研究に基づく40法以上の査読付き論文の発表、並びに心血管系トランスレーショナルサイエンスを推進するための新しいパラダイムの構築に大きく貢献してまいりました。また、Journal of Pharmacological and Toxicological Methods の編集者であり、20以上の査読付き科学雑誌の査読者や、さまざまな科学機関、財団の審査員も務めておられます。また、CiPA イニシアチブのイオンチャンネルグループの共同議長を務めております。それではこれからプレゼンテーションに入りますが、約40分のプレゼンテーションの後に質疑応答の時間を設ける予定です。ただちょっと急遽、ナジャ先生が、参加できるかどうか、緊急な要件が生じたということで、分かりません。もし、本日 Q&A 実施できない場合は、後日個別に対応させていただきたいと思いますので、何卒ご了承いただければと思います。それでは、プレゼンテーションをご覧ください。Now let's get started today's presentation.
Takashi-san, thank you for the introduction. It's an honor to present today's webinar to Japanese colleagues, and I would like to thank every one of you for your attendance. AnaBios is a contract research organization, and we are specialized in human translational research. The human ex-vivo models we have been developing allow drug discovery programs to study drug effects and identify effective therapies. The models help to define drugs that work in a human. They also help to identify patient population, and also they help to provide accurate prediction of dosing in patients. Also, our models can be used to identify safe therapies. They help to define safety margin, and of course, they help to select the dose for first-in-human studies. To have access to ethically consented organs for research, AnaBios works with organ procurement organizations in the United States. The critical elements of our procurement success are: our methods are consistent and we apply SOPs, prevention of tissue damage by avoiding ischemia and reperfusion injury. We have access to large networks that ensures a high volume of organs. And finally, we have access to extensive annotation for each organ. We procure hearts, human DRGs, human spinal cords, lung, and livers. For all these organs, we not only have models that allow to profile drug activity, but also we have biobanking of cell tissue samples to run omics analysis. Next slide demonstrates the quality of tissue samples we provide for omics analysis. Real scores of our tissues are much higher compared to those obtained from tissues provided by other suppliers. For today's webinar, I will focus on the heart models. However, we will have upcoming webinars about the models we have been developing with the other organs. Let me now share the agenda of the webinar. I will provide an overview of AnaBios human technology. Then I will present the human heart models we have to support drug discovery. After that, I will present efficacy studies to demonstrate the utility of our models to support programs targeting treatment for obstructive hypertrophic cardiomyopathy, heart failure, and atrial fibrillation. Then I will present data on how our models can be used to determine the potential of novel drugs to be associated with contractility risk and pro-arrhythmia risk. Then I will provide a quick overview of new models we are currently developing. Finally, I will propose the take-home message. Next slide presents AnaBios models for normal donor hearts. First, note that AnaBios has worked on over 1,500 human hearts the last 10 years, with a rate recovery of 2 to 3 hearts per week. Cell-based and tissue-based assays have been developed. The cell-based assays are employed to support optimization of drugs in early discovery, while the tissue-based assays or models support nomination of drug candidates before entering large animal studies and clinical trials. For tissue-based assays, we have action potential trabecular model to assess potential effect of drugs on the ECG. Second tissue-based model is force of contraction measurement from ventricular and atrial trabecula to assess the potential of drugs to modulate heart contractility. The third model is frequency of spontaneous firing of action potential from sinoatrial node to assess the potential of drugs to modulate heart rate. The fourth model is vasoconstriction dilation model of coronary arteries to assess the potential of drugs to modulate blood pressure. For cell-based model, we have myocyte contractility model to simultaneously assess contractility as well as anti-arrhythmic effect or drug-induced pro-arrhythmia risk. Second model measures intracellular calcium dynamics from myocyte to assess if the mechanism of action of drugs are calcium dependent. Third model measures ion channel recording from myocyte to assess the potential of drugs to modulate the function of a human heart ionic currents. Like the calcium imaging model, this ion channel model can provide mechanistic information. The fourth cellular models measures action potential recording from myocyte to assess the potential of drugs to modulate the myocyte electrophysiology. This slide highlights AnaBios' ability to also recover diseased donor hearts. Here also we have developed cell and tissue-based models. We recover hearts of donors affected by atrial fibrillation, hypertrophic cardiomyopathy, coronary artery disease, heart failure with reduced or preserved ejection fraction. The models we have developed so far are force of contraction tissue measurement and action potential and ion channel models that use atrial myocyte isolated from donors affected by atrial fibrillation. High yield of cardiomyocyte can be obtained from donor hearts. As seen in this slide, they are characterized by rod-shaped morphology, clear striation, and importantly, they don't contract spontaneously. This is an indication that they are healthy and represent well the in vivo ability of these cells to contract only if they receive the impulse from the SA node, atria, and conduction system in the heart. Therefore, they required electrical stimulation to contract and relax. To influence optimization of drugs and help exploring structure-activity relationship, a contractility system must have the characteristic we propose in this slide. The system must be easy to use. The system should allow recording from multiple cells in parallel, so a semi-throughput drug testing can be performed. The system should also allow simultaneous assessment of cell health, physiology, and pharmacology. The data analysis should be automated and rapid. A drug concentration effect curve should be completed quickly. The system should be associated with good reproducibility. AnaBios has developed such system, the MyoBLAZER. This slide presents contractility recording from multiple cardiomyocytes using the MyoBLAZER. First, contractility is recorded from 9 cells in parallel using baseline condition. Next, you can see the decrease in contractility that is caused with the application of verapamil, an inhibitor of calcium channels. The analytical tool we have developed allows to analyze a run of multiple cells recorded with the MyoBLAZER in few seconds. The tool measures parameters that represent the systolic and diastolic phases of the cardiomyocyte contraction. When using cluster analysis, effects of drugs on the kinetics of contractility transient help to fingerprint compounds with inotropic effect. In these slides, we show the multiparametric profiling we published with 26 drugs and the ability of this approach to propose mechanism of action. The same data, when using principal component analysis, can highlight which compound can act via increasing or decreasing of intracellular calcium. All this data are explained in details in our scientific report paper. Now, I will present how our heart models can be used to support projects aiming to treat obstructive hypertrophic cardiomyopathy and heart failure. Data presented in this slide show how human cardiomyocyte and the MyoBLAZER can be used to explore structure-activity relationship with myosin ATPase inhibitors like blebistatin and 3 of its derivatives, BTS and mavacamten. Data indicate that blebistatin, hydroxyblebistatin, BTS, and mavacamten share similar ability to reduce cardiomyocyte contractility. However, para-aminoblebistatin and aminoblebistatin show different contractility profiles compared to the other 4 inhibitors. Also, data with BTS and mavacamten demonstrate low intra and inter-heart variability. Here is another set of MyoBLAZER cardiomyocyte contractility data that again demonstrate low inter-donor variability. And the effects of these drugs on contractility were found to be largely independent of isolations. Data were obtained with dofetilide, a hERG channel blocker, with application of extracellular calcium, with ISO, an agonist of beta-adrenergic receptors, with OMECAMTIV mecarbil, an activator of the myosin ATPase. This compound is in clinical development for the treatment of heart failure with reduced ejection fraction. Also, we obtained data with mavacamten, an inhibitor of the myosin ATPase, a compound recently approved by the FDA for the treatment of obstructive cardiomyopathy. And finally, we obtained data with verapamil, the inhibitor of calcium channels. As indicated earlier, the MyoBLAZER allows to derive kinetic data from the contractility transient. This slide shows that treatment with OMECAMTIV can modulate the kinetics, like rise time and time to 90% relaxation. And such modulation was seen in cells isolated from 4 different donor hearts. However, application of extracellular calcium does not affect the kinetics of the contractility transients. Considering OMECAMTIV and extracellular calcium data, it can be suggested that modulation of the contractility kinetics can be drug-specific. Like the MyoBLAZER, and as shown in this video, our calcium imaging model allows to record calcium transient from multiple cells in parallel. Like the MyoBLAZER, we can analyze the kinetics of the calcium transient signal. For example, we measure peak height that represents the calcium release from the store. And also we measure the peak width at 50% peak height that represents the decay of the calcium signal and its reuptake to the stores. Data indicate that mavacamten, also known as MYK-461, had no effect on calcium transient. In contrast, verapamil inhibits the calcium signal. This slide presents the average data obtained with blebistatin, its three derivative, BTS, mavacamten, and OMECAMTIV. Data clearly indicates that myosin ATPase modulators show no ability to modulate calcium dynamic in a human cardiomyocyte. As expected, the positive control ISO and verapamil modulates the calcium signal. While ISO increased the calcium signal, verapamil caused a decrease in the calcium signal. When exposed to vehicle, our data demonstrate good stability of the calcium signal. And again, the signal was increased with application of two other positive controls, milrinone, a PDE3 inhibitors, and levosimendan, a calcium sensitizers and a PDE inhibitor. Next few slides will summarize the effect of omecamtiv and mavacamten on force of contraction measured in tissue obtained from donors affected by heart failure. As shown in this slide, omecamtiv increased the developed force and relaxation time in donors affected by heart failure with preserved ejection fraction. Omecamtiv effects on the force and relaxation time were not seen with the application of N106, an activator of the circa-2 pump. Note that circa-2 pump plays an important role in transporting calcium from the cytosol back into the stores. In donors affected by heart failure with reduced ejection fraction, omecamtiv showed a higher ability to increase the force when compared to N106 and caffeine. Note that caffeine enhances the release of calcium from the stores and inhibit PDEs. Same ranking of effects can be seen when looking at the effect of this treatment on the relaxation of the contraction represented in this slide by the slope fall. We also tested mavacamten in HFrEF and HFpEF donors. Data indicates that while mavacamten had no effect on the contraction kinetics in normal, HFpEF and HFrEF donor hearts, it decreased the force in all three donor types. Also, its decrease of contractility was much higher in normal and HFpEF compared to HFrEF condition. This may indicate that mavacamten is a better treatment to control contractility in the case of HFpEF versus HFrEF. In the next two slides, I will present another example on how contraction data from our HFrEF tissue donors can support the development of a novel therapy for HFrEF patient. The discovery team at Arena Pharmaceuticals has data demonstrating that overexpression of the beta-3 adrenergic receptors in HFrEF patient represses the contractility. And inhibition of this receptor may increase contractility and thus providing a novel treatment for patients suffering from HFrEF condition. The company developed a selective inhibitor of the beta-3 adrenergic receptors called APD-418. Arena Pharmaceuticals used our heart models to test the novel inhibitor. As seen in this slide, the overexpression of the beta-3 adrenergic receptor was confirmed in HFrEF donor hearts compared to normal donor hearts. Also, data demonstrates that responses to ISO and norepinephrine were augmented in the presence of the inhibitor. Note that the FDA granted Fast Track designation to APD-418, and the compound is now being evaluated in clinical trials. Next few slides, we will demonstrate how our models can be used to support the development of new anti-arrhythmic and anti-atrial fibrillation drugs. To validate our models, we first assessed their ability to demonstrate that inhibition of the late cardiac sodium current can be a promising therapy to treat long QT syndrome arrhythmia and atrial fibrillation. The full validation is detailed in our recent publication in scientific reports. Stimulation of the late sodium current with ATX2 mimics long QT syndrome type 3. As can be seen in this slide, ATX2 application increased ventricular cardiomyocyte contraction and caused arrhythmia. These effects were significantly attenuated with inhibitors of the late sodium current, like ranolazine and GS967. These compounds were developed by CV Therapeutics and Gilead. Also, we found that GS967 acts as an anti-arrhythmic when long QT syndrome type 2 was mimicked with the application of E4031 in ventricular cardiomyocyte. Please note that E4031 is an inhibitor of the hERG channel IKR current. Also, we found that GS967 acts as an anti-arrhythmic when long QT syndrome type 3 was mimicked with the application of ATX2 in atrial cardiomyocyte isolated from normal donor hearts. These slides show arrhythmic events after contraction and premature atrial contraction, measured when recorded contraction relaxation cycles from tissue isolated from donors affected by atrial fibrillation. These parameters are indicative of the atrial remodeling that took place in donors affected by the atrial fibrillation. Inhibition of the late sodium current blocked these arrhythmic events and restored normal atrial function. We used our approach to provide a proof of concept to a discovery team that a novel drug X can have the potential to treat atrial fibrillation. As shown in this slide, we demonstrated that drug X not only prolonged action potential repolarization in atrial myocyte isolated from donors affected by atrial fibrillation, but also drug X inhibits the remodeling-induced arrhythmic events and restored atrial function. Also, the compound inhibited atrial contraction. In conclusion, our data demonstrate that this drug X has the potential to control the rate and rhythm of the AFib atria. Then, the atrial selectivity of drug X was demonstrated by its inability to affect action potential repolarization in normal atrial cells, also by its inability to modulate ventricular action potential, ventricular contractility at both myocyte and tissue levels. Finally, the compound also showed no potential to cause ventricular arrhythmic events. Our data played an important role in the decision-making to prepare IND application for this drug in 2023. I would like to use the next two slides to make the point that we will soon be able to provide explanted hearts for drug discovery. These slides gives you an idea about the main categories of explanted heart we will be able to provide. Next slide provides more details about the subcategories when it comes to cardiomyopathy and congenital heart disease that we will be able to provide. Next, I will present how our cardiomyocyte model can be used to assess the potential of novel drugs to cause pro-arrhythmia and/or contractility risk. This slide is to remind you that the pro-arrhythmia is caused by electrical and mechanical abnormalities, and it is not only a pure electrical ECG problem. When compared to normal hearts, clinical data shows that hearts with long QT syndrome showed not only abnormal ECG, but also abnormal secondary contraction. The tight in vivo relationship between ECG and mechanical function of the heart is also seen at the level of cardiomyocyte. This relationship results in the excitation-contraction coupling we have between the action potential and contractility of cardiomyocyte. Thus, we wanted to use this relationship to simultaneously predict the potential of a drug to cause pro-arrhythmia and contractility risk in one experiment using the MyoBLAZER. And therefore, we wanted to replace the assessment of these two risks by separately testing the drug in two separate cardiac models. To predict pro-arrhythmia, we will measure the incidence of the secondary contraction, i.e. after contraction. To predict contractility risk, we will measure the effect of a compound on the contractility amplitude, i.e. change in sarcomere length. This slide shows data on how, using our approach, we can differentiate the pro-arrhythmic potential of dofetilide, a selective hERG channel blocker, from the safety of verapamil, a multi-ion channel blocker. Dofetilide caused incidence of after contraction, while verapamil only decreased contractility with no manifestation of after contraction. To validate the clinical relevance of our cardiomyocyte contractility model to predict drug-induced pro-arrhythmia, we tested 23 reference drugs with known pro-arrhythmic potential. One assessed these drugs at tenfold therapeutic exposure and compared to published data with stem cell-derived cardiomyocyte. Our model showed a high sensitivity in predicting the pro-arrhythmia risk. The limited ability of phenotypic stability of stem cell-derived cardiomyocyte to predict pro-arrhythmia was observed in recent published data while testing nearly the same reference drugs we assessed in our study. Our data also demonstrate that our human cardiomyocyte contractility model predicts well the non-pro-arrhythmic potential of drugs. Like their limited ability to predict the pro-arrhythmia, the stem cell-derived cardiomyocyte showed limited ability to predict non-pro-arrhythmic drugs. It is also important to mention the challenge of constructing an exposure response in these cells due to the high incidence of quiescence caused by the drugs. As explained earlier, our model, in addition to predicting pro-arrhythmia, it can be used to predict drug-induced contractility risk. This slide shows the contractility data we obtained with the 33 reference drugs we tested. From the data, IC50 potency data and safety margin can be calculated. This data can be used to rank compound and also can be used in decision-making about stopping or progressing a compound to next discovery phase. In the next few slides, I will show few case studies. These slides show data on AZD7762, an anti-cancer drug that was being developed by AstraZeneca. With contractility data obtained from different animal models, it was concluded that the compound has enough of safety margin to be taken to clinical trial. However, a contractility risk was detected in patients at the nearly therapeutic exposure. This effect led to discontinue the clinical development of this compound. When tested in our model, the potential of this compound to inhibit human cardiomyocyte contractility was seen at nearly one-fold above therapeutic exposure. If used in early discovery, our model will ensure that rate of false negative outcomes will be better controlled compared to data derived only from animal models. This slide presented the data of AZD9291, a FDA-approved cancer drug. Our model data suggests that the drug is free of a contractility risk at therapeutic exposures, and only the risk can manifest at exposures well above free therapeutic plasma concentration. Also, our data indicate the low pro-arrhythmic potential of AZD9291. In conclusion, our data suggest a high safety index for AZD9291 and note that neither contractility risk and/or pro-arrhythmia risk have been reported with this drug in patients. In the next few slides, I will show you how our model was used to assess the safety of hydroxychloroquine. During the COVID-19 pandemic, hydroxychloroquine was proposed as a potential safe drug that can treat COVID-19 patients. In collaboration with Novartis, we used our human cardiomyocyte contractility model to explore the safety of hydroxychloroquine. Data indicate that hydroxychloroquine was safe at therapeutic exposure and only caused incidence of arrhythmic events at exposure well above its free therapeutic plasma concentrations. In contrast to hydroxychloroquine, chloroquine caused manifestation of pro-arrhythmia events, and such potential was seen at free therapeutic plasma exposure. Our data correlate well with the clinical data demonstrating the ability of chloroquine monotherapy treatment to cause pro-arrhythmia and torsade de pointe. This slide shows that hydroxychloroquine in the presence of low level of potassium ions, a risk factor with COVID-19 patients, caused the pro-arrhythmia events at therapeutic exposure. We also demonstrated that magnesium can protect against pro-arrhythmia that can be caused by hydroxychloroquine at very high exposures. Also, we showed that fever, another risk factor with COVID-19 patients, did not influence the safety profile of hydroxychloroquine. Next, we tested the combination of hydroxychloroquine and azithromycin. Since this combination has also been proposed as a potential treatment for COVID-19 patients. Azithromycin alone was pro-arrhythmic. Also, when azithromycin at therapeutic exposure was combined with hydroxychloroquine at different exposure levels, this combination converted the safe profile of hydroxychloroquine to a pro-arrhythmic profile that started at therapeutic exposure. When doing the inverse order of combination, hydroxychloroquine kept its safe profile at therapeutic exposures. We next assessed the effect of hydroxychloroquine, chloroquine, and azithromycin in cell line overexpressing the main three cardiac ion channels that control the repolarization of the action potential, i.e. hERG channel, the depolarization of the cardiomyocyte, i.e. NaV1.5 channel, and the Kv1.2 channel that controls the contractility of the cardiomyocyte. Our data indicate that hydroxychloroquine inhibited hERG and Kv1.2 channels. However, chloroquine was found to block hERG channel, Kv1.2 channel, and NaV1.5 channel, while azithromycin showed weak potencies against these three ion channels. This slide shows the cardiomyocyte contractility data we obtained with the three drugs. Data indicates that hydroxychloroquine and chloroquine inhibited contractility, and hydroxychloroquine effect is not influenced by magnesium or potassium ions. Also, our data indicates that hydroxychloroquine contractility risk is not impacted by azithromycin. However, hydroxychloroquine impacted the effect of azithromycin on contractility when the order of the combination was hydroxychloroquine first and azithromycin second. In summary, our cardiomyocyte data generated with hydroxychloroquine, chloroquine, and azithromycin played an important role in guiding the clinical team at Novartis in designing a clinical study. The next few slides provide guidance on how we use our cardiomyocyte contractility model to detect contractility risk that can be caused by the inhibition of the cardiac sodium channel. In our model, inhibition of sodium channels causes incidence of contraction failure, as shown in this slide. If the sodium channel is blocked, the cardiomyocytes can't initiate an action potential, and therefore the excitation-contraction coupling can't take place, and this leads to contraction failure. Contraction failure may lead to calcium overload. Such calcium overload can initiate strong contractions and pause-dependent arrhythmias. This slide shows that contraction failure can't take place when we inhibit the calcium channel. Under this condition, we only reduce the calcium influx while the excitation-contraction coupling takes place with the depolarization of the cardiomyocyte due to the activity of the sodium channels. This is how we differentiate inhibition of sodium channels from inhibition of calcium channels in our cardiomyocyte contractility model. Here is a case study of a project with sodium channel liability. With only 3 iterations of drug testing, the model provided SAR data that helped the project to significantly reduce the sodium channel liability. Based on sodium channel data generated using cell lines, a recent publication demonstrated that a human QRS prolongation occurs on average at the free plasma levels 15-fold below the IC50 at the human NaV1.5 channels. We carried out similar analysis using the IC50 contractility data for 8 sodium channel blockers we tested in our human cardiomyocyte contractility model. As shown in these slides, when comparing the risk window and safety margin between NaV1.5 and contractility data, we found that the risk window and safety margin calculated with our data are better predictors of a QRS prolongation observed in humans. I will now share with you the new human heart models we have been developing in 2021. All myocyte data I presented were generated in AnaBios lab. We want to provide scientists with the possibility of having the myocyte in their lab so they can use them to conduct their own experiment and test their own ideas. Cells shown in this slide are cells that were flown to Florida and then flown back to AnaBios. The shipped cells contracted in response to stimulation, and again, multiple cells can be recorded with the MyoBLAZER. Data demonstrate that increases in contractility induced by iso and milrinone were not different between shipped and non-shipped cells. Also, we noticed that baseline contractility was also not different between the two groups of cells. We hope to start distributing these cells in the very near future. Please note that we are finalizing a collaboration with Professor Dr. Sugiyama at Toho University for the shipped cells to be tested in Japan. The ability of assessing long-term effects of a drug is an important addition to our heart models. To this aim, we have developed conditions that allow to maintain adult human primary ventricular cardiomyocyte in culture with pacing for 24 hours. The cells kept their rod-shaped morphology with a clear striation, did not go through a dedifferentiation process, and displayed a normal excitation-contraction coupling in response to electrical field stimulation. Next, we tested the pharmacological response of this 24-hour cardiomyocyte to 4 reference drugs. We tested responses to dofetilide, to isoproterenol, to ranolazine and verapamil, 2 multi-ion channel blockers. Dofetilide had no major effect on contractility. While iso increased contractility, ranolazine and verapamil were found to decrease contractility. Data also demonstrate that while dofetilide and iso caused after contraction, ranolazine and verapamil showed no potential to cause after contraction, i.e. pro-arrhythmic events. Also, additional testing is underway to further validate these 24-hour models. The model is already being used to support clinical programs. I would like also to mention that we are working to develop the next generation human cardiac model for drug discovery. The vision we have for this model is presented in this slide. We want the model to support both discovery and cardiotoxicity assessment. We want the model to emulate a human cardiac cycle. We want the model to emulate hemodynamic of normal and diseased hearts. We want the model to allow the assessment of drug effect on electrophysiology, calcium dynamics, and force of contraction. We want the model to permit assessment of acute and long-term effects of drugs. We want the model to also permit the assessment of drug effects on transcriptomics, biomarkers, metabolism, and the proteomics. We want the model to be used to assess the potential of novel drug, gene, and cell therapies. And finally, we want to use artificial intelligence and machine learning to integrate the data and inform the best decision making. A final important point about the model is its flexibility to accommodate data generation that a discovery project may need to progress its drugs across preclinical phases with high probability to deliver successful clinical trials. We strongly believe that 3D cardiac slice cultures can be the next generation human cardiac model for drug discovery. As shown in this slide, the MyoSlice is a 6-chamber system that can be cloned into multiple MyoSlices to provide semi-throughput drug testing. The MyoSlice can emulate the human cardiac cycle, i.e. the relationship between pressure and volume that is characteristic for each donor. Slice cultures with the system stay viable for 12 days with maintained functionality and minimal change in gene expression. AnaBios is working closely with Dr. Tamer Mohammed, the investor of the system and a world cardiac slice expert, for transferring the system to AnaBios. We aim to validate the system during Q4 of this year with a plan to start services in Q1 of 2023. I will now close my webinar with the take-home message. Our data confirms the ability of human heart models to detect the potential of drugs to be effective in treating heart disease and to predict drug-induced cardiotoxicity. We believe these models can be employed during early discovery and late preclinical development. They can also be used to generate follow-up data to support the clinical programs. The models can be employed in conjunction with traditional efficacy and safety discovery models. Such combination can provide pharmaceutical companies with unique translational opportunity to rank novel drugs from most to least cardiotoxic, from least to most efficacious, and to well-define human safety and efficacy windows.